Welcome to the annual summit on positive psychology and psychological resilience based in Toronto, Canada. Allow me to start today by asking you a personal question. Have you ever heard this word before? Epigenetics? Probably most, if not all of you, have heard it before. So let me venture with a second question. Could you explain to your colleague, or if you're in the auditorium, to a neighbor on your left or your right, and explain how epigenetics works? Probably the number of hands have now gone down dramatically. And that's good, because that's why I'm here today. My name is Whitney Brer, and I'm a transformational coach, author on the topic of positive leadership, and trainer and keynote speaker in the fields of positive psychology and resiliency. And I'm guessing many of you, like myself, you're passionate about topics like happiness, subjective well-being, and the mind-body connection, right? I'm completing my PhD at Fielding Graduate University in California. I live in Germany, and I'm focusing my research on the topic of sustainable happiness. So what does sustainable happiness have to do with genetics or epigenetics? I mean, is happiness like in our genes? So like many of you in the audience today, I'm guessing that you're familiar with this model, the happiness pie. Right? A model put forward in 2005 by Dr. Sonia Lyubomirsky. And this pie is where my journey in the field of epigenetics began. And it's something I'll touch on in just a few minutes. So, what's in it for you today? Why should you give me your attention for the next 20 minutes? Well, when I finish, you'll understand the connection between this fruit fly, this pie, and this triangle, and what they all have to do with epigenetics and sustainable happiness. So if you're ready, let's embark on our journey today with this seemingly innocent fruit fly and why in the year 1956 it turned the scientific world upside down. Let me introduce someone to you. This is Conrad Waddington. He was a British development biologist who successfully demonstrated that the inheritance of a characteristic acquired in a population of small fruit flies was done so as a reaction to an environmental stimulus. In other words, something outside the fruit fly triggered a reaction in its genetic inheritance. I'm telling you, this was one small step for a fruit fly and one giant leap for genetics, so to speak. Many people scoffed at the idea and claimed that this was an example of phenotypic plasticity. What is that? That's the ability of an organism to change in response to stimuli from the environment. For example, some caterpillars mimic flowers or leaves of the tree depending on their diet. This is often considered an adaptation strategy for surviving in variable environmental conditions. For example, if this caterpillar was green, it would probably have a very short lifespan. Or vice versa, if this one was yellow. Right? Critics claim the fruit flies in Waddington's initial studies didn't change genetically. In other words, they say their DNA wasn't rewired, but rather they simply adapted to the new environment they were subjected to. You know, in 1956, Waddington didn't know exactly how it had happened, but he continued with his studies and could eventually prove his critics wrong. And several years later, after much more experimentation, Waddington introduced a model called the epigenetic landscape. This is how Waddington depicted it originally. So imagine a ball at the peak of a mountain which is let loose. And imagine the ball rolling down the mountain and rolling through kind of a serpentine valley. As the landscape changes, the ball has to constantly make choices regarding which path it's going to take, to the left, to the right, straight ahead. The ball not only rolls through the valley, taking various paths, but it also picks up like debris and stuff along the way, which kind of sticks to it. The rolling ball is Waddington's representation of a cell's development over time. The valley and the ever-changing topography, that stands for the environment that controls these options. The debris, the stuff it picks up along the way, stands for the environmental stimuli it is exposed to over time. Waddington selected for and bred from the animals displaying new characteristics after being exposed to environmental stimuli. These new characteristics were analogous to the traits the rolling ball had picked up while going through the valley as it was being exposed to the ever-changing topography, or in Waddington's laboratory, environmental stimuli. And after just a few generations, he discovered that he could breed from these animals without exposing them to the environmental stimulus. And they would still show the new characteristic or trait. Phenotypic plasticity or mutations, which his critics claim were causing the changes, they take much longer and only happen through multiple generations. Waddington, with the help of external stimuli, had demonstrated these changes in just a few generations. And you know what? He knew he was onto something else. So, since Waddington's original discovery, our knowledge of DNA in the field of genetics has both advanced significantly. Today, scientists, when referring to epigenetics, are referring specifically to heritable changes in gene expression that do not involve direct changes in the DNA sequence. Okay? So this is a quick crash course in the history of genetics. If you're ready, let's turn our attention to explaining how epigenetics even works and what it has to do again with this happiness pie. Simply put, epigenetics describes how DNA interacts with small molecules situated within cells, which can turn on, activate, or silence, deactivate genes. It's as if these small molecules act like a light switch, turning genes on and off. Epigenetic changes can enhance or get in the way of the reading of specific genes. In other words, they can turn on or turn off this reading. The most common way that interference happens is that DNA, or the protein it is wrapped around, and that's shown by the yellow balls in the diagram, gets labeled with some small chemical tags. Some of these chemical tags, like methyl groups, prevent gene expression, causing the DNA to coil more tightly, making it inaccessible. So if this process described is a bit too technical, imagine this. Imagine a soft children's ball. Now imagine taking a string and wrapping it around this ball. The ball is symbolic for the protein, and the string represents the DNA, which is wrapped around it. So what would cause the string to wrap even more tightly around the ball? The answer is chemical tags. And one of these chemicals is methyl. hence the term methylation. Methylation of the gene causes the gene to be silent. It's almost as if the string is being pulled so tight that the gene is being strangled and has no voice. It cannot speak, so to say, and thus it just can't be heard. The gene is still there, and the DNA has not been changed. It's only been deactivated, silenced. It's kind of been strangled. Enhancing description is just the opposite. There are some chemical tags which cause the DNA to unwind, making it easier to transcribe. And this boosts the production of the associated protein. In this case, the string around the ball relaxes, stopping this kind of strangulation process and letting it speak or express itself. Okay? What else is there to know? What else is important? Well, for one thing, epigenetic changes, as Waddington discovered with his fruit flies in 1956, do you know what? They can survive cell division. This is important because this means that these changes can affect an organism for its whole life. There are about 200 different cell types in the human body. That's a lot. For example, we have heart cells and liver cells, we have brain cells, and all of these 200 cell types all have the same genome. But each individual type has its own epigenome. In other words, the genome is kind of like a cookbook for a living organism. Each genome contains a complete set of genetic instructions. Everything that an organism needs to know how to build itself, how to allow for future growth and development. It's all there in this cookbook. So each of these 200 cells have the same genome, the same genetic recipe, but each cell has its own individual epigenome, which is special for that particular cell type. The epigenome, remember, each cell has its own type. It kind of works like a mediator, mediating over the course of a lifetime between genes and their environment. In other words, our body has employed over 200 different mediators, one for each cell type. And you know what? They have lifetime employment in you. And if they had a name tag on their uniform at work, it would say something like, Epigenome Mediator at your service 24/7. They take their job seriously, and they're always on high alert for any chemical tags which come their way, especially those methyl groups which might be sneaking around. The chemical tags can turn these genes on and off. And now, this is important because this can be influenced by environmental factors, like diet, chemical exposure, social experiences, even the people around you who may be toxic for you, medication. And these changes which result from these environmental influences can eventually maybe lead to disease if they, for example, turn off a gene that, for example, makes a tumor-suppressing protein. Thus, adages like, you are what you eat, you're the average of 5, you are what you think. You know, these aren't as philosophical as they may initially sound. So now let's do a time warp from 1956 to 2005. In the time span of half a century, what does all this have to do with happiness and genetics? Although the concept of epigenetics caused quite a stir when Conrad Waddington first published his findings in the mid-1950s, it became an even more heated debate in 2005 when Dr. Sonia Lyubomirsky proposed the happiness pie. She and her colleagues set out to explore this question. Through what mechanisms, if any, can a chronic happiness level higher than the set point be achieved and sustained? Now here's a quick definition. A chronic happiness level is not about being in a permanent state of bliss or scrutinizing single changes in one's happiness. A chronic happiness level is about looking with hindsight over the last few months at one's own perception of overall happiness and deriving a mean. Although at first sight, happiness and epigenetics may seem like two vastly different topics, they're not, according to Lyubomirsky. This model ascertained that an individual's happiness is comprised of three factors: 40% intentional activity, 10% life circumstances, and genetically predetermined set point, 50%. It is this assumption from Lyubomirsky that 50% of our happiness can be traced to a genetic set point that caused quite a stir. So what's happened in the meantime? Well, 15 years later, in 2019, 2020, Brown and Rohr critically reevaluated her calculation and proved it to be problematic, if not simply pie in the sky. The major fault of this happiness pie and its calculation was based on something that's called variance decomposition. Now, the underlying problem with using variance decomposition to predict an individual's level of happiness is that this model can only be used for whole populations. And the happiness pie applies to an individual subject level. Critics go on to argue that the variance decomposition model is only accurate when all three factors, intentional activity, life circumstances, and a genetically predetermined set point, do not interact and are independent of each other. And Brown and Rohr rightly argue that this is just not possible. Another problem with this happiness pie is that it's based on three components. And this automatically assumes that these are the only possible causes of variance in happiness. For example, circumstances, which makes up 10% of the pie, is kind of a catch-all term to include all the vicissitudes of life that are outside an individual's personal control. And this omits a multitude of various factors, leaving the model with no wiggle room for error. So, that being said, is this whole thing just pie in the sky or not? Should we just throw in the towel and give up? Is happiness a lost cause? I argue, nope, it's not. Dr. Sonia Lyubomirsky argues that the controversy over these exact numbers and the composition of the pie is detracting from what she and her team were originally investigating. Once again, through which mechanisms, if any, can a chronic happiness level higher than the set point be achieved and sustained? How much or what percentage of your set point is 50%, or is your set point 37.9, or is yours 53.7? We'll probably never have the answer to that. We may never know how much exactly makes up intentional activity. 40%, 56, 23. But as I say never. But one thing for sure that we know that intentional activity can boost our happiness and improve our subjective well-being. And happiness does seem to be a topic that a lot of people are interested in. The World Data Bank of Happiness, for example, has grown to include nearly 40,000 findings, with another 20,000 research findings waiting to be entered, research findings. I mean, that's a whole lot of scientific interest in happiness. Now, two questions have shifted into focus. What interventions or mechanisms are there that can help boost happiness? And how can we sustain that boost in our happiness? In other words, how can we sustainably move the dial on our own set point just up a few notches? And does this mean that we are striving to stay in a permanent state of bliss? Does it mean that we have to wear rose-colored glasses in life? No. In fact, negative situations can help us truly appreciate and savor the good times in life. And this brings me to the third picture for my introduction to this triangle, more specifically called the Pinrose Triangle, which represents an appropriate analogy for the sometimes ambiguous link that exists between scholars and scientists on the topic of epigenetics. The triangle allows the pictures to flip back and forth equally between possible perspectives, all depending on the viewer, your point of view. In this picture, the question of epigenetics, or a set point, is being examined through three perspectives: the medical experts, the mind-body community, and positive psychologists. While the medical community is exploring cures and treatments for cancer, for example, type 2 diabetes, and autoimmune diseases, and measuring changes in methylation and epigenetics, the mind-body community, for example, is studying interventions like meditation, hypnotherapy, guided imagery, and yoga. Positive psychologists are focusing on developing, testing, and measuring the effectiveness of interventions or intentional activities which boost happiness, striving to have a long-term impact on an individual set point. Such interventions have, for example, focused on the link between subjective well-being and gratitude, hope, joy, purpose in life, character strengths, the PERMA model, P-E-R-M-A, resiliency, optimism, as well as depression and mental illness, just to name a few. Although these approaches may at first sight seem to have little in common with each other, these three communities, they actually have a lot in common, because these three approaches are at a crossroads, and they offer a unique opportunity to synergize their knowledge. For example, a 2021 study of breast cancer survivors showed that spiritual well-being can ward off depression and improve quality of life. Another scientific study showed that social well-being improved the mental and physical health of women who had just been diagnosed with non-metastatic breast cancer. In this study, social well-being led to reduction of inflammatory signaling of pro-metastatic leukocyte gene expression. Why not contribute knowledge of interventions concerning spirituality and social well-being to cancer researchers? In another study, neuroscientists proved that stress reduces the size of the hippocampus. Here's a fantastic opportunity to contribute interventions which reduce or prevent stress. A further study showed that spiritual well-being and hope improve the mental and physical health of patients with cardiovascular disease. And one last example. The results of a further study done in 2021 show that love, curiosity, persistence, hope, and zest are strongly associated with the subjective well-being, especially in conditions such as quarantine and isolation during COVID, and can support specific aspects of it. All of these studies have one thing in common. They have all shown the connection between the medical researchers, the mind-body community, and positive psychologists. And this connection is not as ambiguous as it may seem at first. In fact, the connection is crystal clear. Is our destiny completely in our genes? No. Is the genetic set point the same for each individual on the face of this planet? No. Does the ongoing research offer hope for the future? Yes. And on the closing note of hope, hope is one of the 10 positive emotions from Barbara Fredrickson that gravitates around positivity. You know, hope forecasts possibilities and helps us see it doesn't matter if the glass is half full or half empty. What matters is that we know how to refill our glass. And from my point of view, this is the quintessence of the field of epigenetics. Knowing there are ways to improve our well-being, our happiness, and yes, have a positive impact on our epigenetics. And this fills me with hope. Thank you so much.